Hello, everyone, and welcome to today's webinar, Exploring the Clinical and Immunopathological Association between Long COVID and Sjögren's disease in mice and humans. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's presentation is interactive, and we encourage you to participate by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. Also, if you experience any technical questions, you can utilize this Q&A box and we will assist you directly. Today's educational web seminar is presented by LabRoots and it's brought to you by Olink Proteomics. To learn more, please visit olink.com. I would now like to welcome our esteemed speaker, Dr. Cuong Nguyen, Associate Professor, University of Florida. For a complete biography on our speaker, please visit the Biography tab at the left of your screen. Welcome, Dr. Nguyen. You may now begin your presentation.
Well, thank you, Christy. I first want to thank Paula Parent and Raza Ahmed at Olink for setting up this meeting. I also want to thank Christy for her technical support here. This is my first webinar, so I hope things will go smoothly. I do want to thank the attendants for spending your time and with me today. So for some of the learning objectives today I want to outline the clinical symptoms observed in COVID-19 patients and its similarities with those seen in Sjögren disease patients. I want to discuss the outcomes observed in models infected with SARS-CoV-2. Also highlight the prospective advancements in diagnosis and treatment of these conditions. So I think by now, COVID-19 has affected about 1 billion people in the world today. And I'm sure that number is a huge underestimate. So in the USA alone, 3 out of 4 adults over the age of 16 are infected with SARS-CoV-2. So since day one of this pandemic, every number is just crazy and staggering. So as you know, a typical course of a SARS-CoV-2 infection, a COVID-19 infection, is an acute phase of infections, right after exposure of SARS-CoV-2, and then you have an ongoing symptom that lasts about 4 weeks, unless you have school kids like me, where I have the pleasure of being exposed to different variants of SARS-CoV-2. So my symptoms go on and on forever. So after 4 weeks, you start experiencing something called long COVID or post-acute sequelae. So this is when it's have not resolved, and now it's actually become systemic, where you're affecting multiple organs in your body, from the lungs, the digestive tract, the heart, the brain, the liver. And we speculate a few mechanisms for this. It could be the viral reservoir of persistence, and for us, for Sjögren, is the reservoir in the lacrimal gland and salivary glands. And then you lead to this tremendous inflammation, uncontrolled inflammation, and some patients experience autoimmunity, and then you have the microbiome dysbiosis. So this is a brief explanation of how it's actually become multi-system symptoms along COVID, anything that happened post 4 weeks post-infection. And I want to show you some of the clinical symptoms that are associated with this. One is the mental health, right? Depression, anxiety, sleep problem. And you see respiratory problem, you see gastrointestinal problem, you see musculoskeletal system, joint pain, myalgia. You will see neurological system defects, cognitive defects. You see loss of taste and smell. These are things that we heard a lot when you experience in COVID-19 infection. Then you have fatigue, body aches, fever, chill. A lot of patients, about 10.6% of patients actually experience dry mouth, and then 75% of patients experience dry eye. So, and I want you to look at the part where I circle red there. This is the symptom that the patients with Sjögren's actually experience when they have Sjögren's. especially with the joint pain, the myalgia, the dry mouth and dry eyes. This is a symptom they actually experience. So you see there are a lot of similarity between COVID-19 patients, especially long COVID patients with Sjogren syndrome, Sjogren disease. So what is Sjogren's disease? Okay, so the main organs that are affected by Sjogren's are the lacrimal gland, which lead to keratoconjunctival sicca or dry eyes. And then you have xerostomia, which is dry mouth. So the symptoms are so painful that imagine you're on the beach and you get sandy eyes and you cannot clear that sand because you have no eyes and no tears in your eyes to wash it off. So this is very painful. In the USA alone, you have about 4 million Americans affected with Sjogren's, and that there's a 2.5 million patients are underdiagnosed. It's mainly a woman disease where 9 out of 10 of patients are female. Um, it takes a long time to diagnose. This is 3 years to diagnose with Sjogren. And when I started out in this profession, it would take 10 to 15 years. So the diagnostic has improved significantly. And it usually occurs in postmenopausal women, but it can increase with age. So this is an example that I mentioned earlier about some of the systemic extraglandular mutation with Sjogren disease. So again, it's not just affecting the lacrimal gland, the salivary gland that leads to dryness in the mouth. You have a systemic extraglandular manifestations. Here, I show you a few examples here. The musculoskeletal manifestations. You have myalgia, morning stiffness. You have dry cough. Um, you have nausea, dyspepsia, um, 10% of patients with Sjogren actually develop renal diseases. You experience vasculitis. And then last is that about 48% of patients actually develop non-Hodgkin lymphoma, um, which can actually quite deadly. Not just affecting quality of life, but now you have developed B-cell lymphoma, which can be detrimental to your life. So, in terms of how we actually diagnose Sjogren, and the classification has changed over the time since I've been around. Uh, so now the latest update is that it based on 5 criteria to diagnose Sjogren's. Okay, and you can see that it's weighted. First of all, it's based on the labial salivary gland biopsies, right? And you can show in the bottom figure there where you see the minor salivary biopsies histology there with the staining of B and T cells present. If you're positive, you have a score of 3. They look for anti-SSA or Rho antibody. If positive, score 3. Ocular staining, you can see the lissamine green, a fluorescent dye green. If they're positive, score 1. The Schirmer test is measure how dry your eyes is, and if it's less than 5 minutes per 5 millimeters per minute, where they put a thread right there, you have a score of 1. Then you can do an unstimulated saliva flow rate of less than zero point 0.1 millimeter per minute, score of 1. So you add up the score. If you have a score more than 4, then you consider to have Sjogren. So if you add up all of this, if you have more than 4, you will have uh, classified as Sjogren. So what the challenge is with human patients that you can really understand disease at a chronic or clinical stage. You don't get to understand the preclinical stage, what happened prior to disease onset. So because of that, for the last, I would say 10, 20 years, my laboratory has developed many different models, mouse model to look at different um, etiology, different factors that show Sjogren. So we learned one thing that the NOD mouse model, which is the non-obese diabetic mouse, okay? So this is a mouse that have for a long time been used for study Taiwan diabetes, right? When we look at the lacrimal and salivary gland, we look at the secretion function of these two organs. They actually have infiltrates in the salivary gland, the lacrimal gland. They actually lose saliva secretion and tear secretion. They have a lot of autoantibodies in the serum. So this is a great model for Sjogren's, but also with Taiwan diabetes. So this is so it's not a good model to really look at just primary Sjogren's syndrome where you just have dry and dry mouth. So because of that, we rely on the NOD B10H2B mouse. So this is when we replace the IAG7 MHC of the NOD with a different with the C57 black C57 black tan um, HOA, where now the NOD B10 mouse only have Sjogren's and not diabetes. Okay, because of that model, we can actually make a SCID mouse would have no functional B and T cells, and because of that, they have no Sjogren. We actually knock out the IgMu, which is knock out the B cells. They have T cell infiltrates, but no clinical disease, right? No B cells. We knock out the STAT6 molecules involving the classic switching from IgG to IgM to IgG1 antibodies. They have no disease at all, right? We knock out the IL-4, which involve in proliferation of T cells, okay? And now they have no Sjogren. We look at, you use GFP as a Doppler transfer experiment to look at different cell types. So because of that, it's very, for us, very useful to use mouse model to look at different aspects of Sjogren. So the one thing we also do is that the NOD mouse doesn't have a good control mouse, healthy control. And we all, as we've done a lot of mouse work here, the C57 black 6 is a perfect mouse control for a lot of diseases, a lot of model we study or anybody study. So we learned that there are mouse strains that with the B6 background with different NOD locus or loci on those mouse, right? Here example that a mouse established called NOD IDD5 mouse. Okay, so this mouse on the chromosome of 1, it has a chunk of the IDD5 locus on that mouse. Okay, this mouse here did not get Sjogren, but they get a lot of they call pathological changes in the salivary glands. Right, they have delayed organogenesis. They have a few strange things happen to the salivary glands, to the at uh, uh, the cellular levels, but they don't have the full blown Sjogren's. Right, they have no antibodies, they have no infiltrates. Then we look at another mouse model where there's an IDD3 locus on chromosome 3. Okay, so this mouse here have normal pathology of the glands, but they have infiltrates. They have infiltrate in the glands, but they don't lose the saliva secretion. So we thought if we bred them together, where we create this congenic strain with two of those IDD5, IDD3 loci, right? Or we call them AC1, C2. This mouse here with the two loci, develop the whole spectrum of disease where they have the pathological changes in the gland and also they have the immunological changes in the gland. And then they actually have lost the salivary secretion over time. And then when we compare the our mouse strain with human pathology, human disease, the different parameters there, you can see we compare dacryoadenitis, salinitis, pro-inflammatory cytokines, autoantibodies, uh, KCS. We compare decrease in saliva flow rates, and you can see on the right column, they are very, very comparable, showing that our model actually mimic the human disease quite well. And then having all these models, we actually created a disease profile process where we learn from the beginning of disease all the way to the clinical onset disease. So we divide the pathology development of Sjogren into three different phases. Okay, these are continuous but distinct phases of Sjogren. From 0 to 4 weeks of age or 0 to 8 weeks of age, we know there's delayed organogenesis. So if you look at the formation of a salivary gland, it looks like a grape-like structure. But in our mouse model of Sjogren, you see less grapes looking when we look at the salivary gland, right? So we live as a delay in organogenesis. We see the aberrant gene and protein expression. For example, we see the expression of a submandibular gland protein in the parotid, we see the expression of parotid protein in the submandibular gland, okay? And this is the early stage where we start seeing the actin cell apoptosis. And at phase 2 here, because of the inflammation induced by apoptosis, we see the influx of locus infiltration in the gland. And with that, at phase 2, from 8 to 12, 16, we see production of antibodies. We see the parotid secretory cleavage, right? And we have used it as a marker for detecting Sjogren. Especially at this stage here, we start detecting the presence of muscarinic excitatory receptor. So think about when you smell pizza, right? You release acetylcholine that bind to the M3R. So the idea is that in patient with Sjögren, you have antibodies that bind to the acetylcholine receptor and block the signal transduction. And because of that, you cannot secrete saliva. And then at phase 3, from 20 and above, you start seeing the actual clinical disease, the loss of saliva flow rate. So we have developed this model about 10 years ago, where we start seeing that the initial insult of the gland could be a viral component, could be the apoptosis, right? That start produce interferon gamma, okay? That would activate the macrophage, activate the epithelial cell to recruit, to activate VCAM and ICAM to recruit a particular subset of cell called Th17 cells that produce IL-17. They come in, and then now you have Th2 cell that produce IL-4, the Th17 cell produce IL-21 that involved in the switching IgM to G1 antibody, which is pathogenic. You also have Th17 cell produce IL-23 that involved in the maintenance of Th17 cells, okay? And that produce a lot of inflammatory cytokines and MMP, which allow for the destruction of the glands. And because of that, you have essence cell destruction. So we developed this model, like I said, about 10, 15 years ago when I was a young, handsome postdoc. I mean, some of it's still true. I mean, not the handsome young part, but the model is still true in the sense that that we know IL-17 is involved in this. We know Th17 is involved. We know Th1 is involved. We know Th2 cells involved. But we wanted to understand the complexity of disease and how we can expand our understanding in terms of learning more about the cytokine, the chemokine expressions in the mouse model, especially at the different stages. Okay, so because of that, excuse me for a second, we approached Olink, which created developed the target 48 mouse cytokine panel. Okay, and the reason why we picked this target panel is that it has, I think, about 45 different chemokine cytokines in that panel. And we have frozen samples, precious frozen sample that we have to stay over time and require very small amount of volume that we can use for the assay. We use up to 1 microliter to run the assay. So we decided this is a great way to approach this. The best part is that we actually, having this many cytokine chemokines, we can actually extrapolate the pathway that involved in the disease pathogenesis. So but the assay is also quite simple in the terms that it's a simple assay where they have desired oligonucleotide that conjugate to a antibodies of interest of your cytokine, and then there's a PCR reporter, right, that can amplify that. So using the PCR signal, they can extrapolate to actually concentrations in the sample. So that is quite easy to do. Um, as you can see here, we actually look at the C57 black 6 mouse, which is a normal mouse. We look at young mice and old mice, right, that supposedly have no disease at all, young and old, maybe some changes there. Then we use the AC1 and AC2 mice, right, from young, which is no disease, and then older mice, which we know have clinical disease. And we compare those 4 parameters in this using this panel. You can see here, you can see the upregulation of CCL2 at the older shown in the mouse. You see expression of CCL4. You see upregulation of CCL11, 12, 17, and 22, right? And then you look at literature here, all of this actually have been shown to be upregulated in human with Sjögren. So clearly there's a correlation there that we can extrapolate. Then we look at chemokine ligands. We look at the colony stimulating factor 1, 2, 3. We look at CXCL1, 2, 9, and CXCL11. And when you focus on CXCL9, this is actually highly upregulated in the Sjögren's mouse. This is a chemokine involved in the germinal formations in the salivary gland. And you can see in patients with Sjögren, 30% of patients with Sjögren develop germinal center in the salivary gland. So this is quite indicative of what happening in the patient. And then we look at cytokine levels here. I want to focus on interferon gamma. It's also upregulated. IL-1 beta, IL-2, IL-3, IL-4, IL-6 are all upregulated in the disease mice. And then now we look at other cytokines, particularly one involved in Th17 cell differentiation. And you see increase IL-17A, F, IL-21. You see the increase in TNF alpha, right? So these are molecules, and then we learn different things. IL-16 involved in recruitment of memory T cells, and that have not been shown in Sjögren, right? So this is fascinating because if you look at the older mice, older Sjögren mice, they're actually quite elevated. The mouse is quite staggering at about 5,000 picogram per liter. So that's a lot. And then we look at the regulatory cytokines, right? We look at CTL4, which we know that compete with CD80 to suppress the T cell response. We see production of IL-10, which involves regulatory T cells, IL-27, which inhibit IL-17, right? And we see the CD274, which is PDL1. They all upregulated in the older mice, in the older Sjögren mice. So what does it say? Like, you want this to be upregulated, but it's not somehow defective or dysfunction in suppressing immune response. So there must be some sort of a immune dysregulation of Sjögren that we need to learn more, or we have already learned some of that processes. So using those data, I have done a cake analysis pathway. You can see when we put this, only the proteins, the cytochemical is upregulated in Sjögren and put into the data analysis. And you can see that you see the increase in our 17 pathway, JAK-STAT pathway. You see the inflammatory bowel disease pathway. You see viral protein pathway involved in this. And then further, we actually look at the protein that the chemokine, cytokine that overexpress in Sjögren mouse. Are they involved in other diseases where we look at biological disease associated pathway? As you can see here, it's involved in arthritis, lung disease, eosinophil, involving common cold, allergic hypersensitivity diseases. So they're actually very distinctive pathway across over with other diseases that we see in Sjögren. So in summary, having a comprehensive chemokine and cytokine panel allows us to explore some of the established pathway, also novel pathway that haven't been seen in Sjögren. We also identify potential target using this, right? We understand what recruit lymphocyte into the thyroid gland, what chemokines are being overexpressed. We actually can determine how we can block that, right, to prevent destruction by the immune cells. The most important thing we learn is that there are actually many cytokines that it's also mimicking the long COVID-19 that we will discuss a little more, especially the expression of interferon alpha, especially the expression of interferon beta, IL-1 beta, IL-6, TNF, IB10, and CCL11, which is a chemokine involved in cognitive defects in patients with COVID-19. So there's a lot of crossover between SARS-CoV-2, COVID-19, and Sjögren that I think we need to explore further. And one way we do this is like we actually look into this to see how it can SARS-CoV-2 induce Sjögren disease. One thing we look at, okay, so we learned that the H2 express everywhere, right, in a lot of places in the tissues, and involved in the receptor of a spike of the SARS-CoV-2. So we want to see if are the expression of H2 using the genetic human H2 mouse in the transgenic mouse. And we look at especially the salivary gland and lacrimal gland, in male and female. As you can see that in the lacrimal gland, there's tremendous expression of ACE2, much higher than the expression of ACE2 in the salivary gland, right? So clearly indicate that although these are systemic transgenic mouse with ACE2 supposed to be everywhere, you can see that they actually express in the salivary gland and the lacrimal gland. And then we want to see, what we do is we actually infect mice with SARS-CoV-2, and we use a Wuhan strain, very beginning, the Wuhan strain. We want to see how long do they persist in the gland, and we want to see if they actually stay in the salivary gland or lacrimal gland. And you can see that we infect mice in the 860 PFU of the Wuhan strain, and we euthanize the mice 21 days after post-infection. And you can see that when we stain for the lacrimal gland and the salivary gland, that you can see the nucleic capsid protein of SARS-CoV-2 in those organs. And in the last figure, we see a little dot that shows the actual cells that are infected with SARS-CoV-2. Then what happened was, when we infect mice with SARS-CoV-2, we want to see how does it change the salivary gland function, right? Secretory function of the salivary gland. You can see here in panel A, you start seeing that the health control mice which infected with PPS, with infected mice are infected with SARS, the Wuhan strain. You can see a significant decrease in the saliva flow rate in those mice, and that correlates to about 50% loss in saliva in B. And then C, you can see that infected mice has some changes in the weight, but it's not statistically significant. Then we look at the pathology of the salivary gland and the lacrimal gland, and you can see here in A, where you see in the control mice, the salivary gland looks pretty normal, right? If you look at the infected mice, you can see those dark little circle little staining there. Those are lymphocytes, right? Infiltrates in the salivary gland, and we look closely to those epithelial cells. We can see that there's apoptotic bodies in the ductal cells of the salivary gland, right? So when we look at the control lacrimal gland, we see the same thing, that you see all the little dark blue staining there. Those are infiltrates in the salivary gland, and close examination, we can also see the apoptotic bodies in the lacrimal gland. And then we quantify that, right? So we know that all the mice infected with SARS-CoV-2 have inflammation in the lacrimal and salivary gland. The question is, do they actually develop a focus score that we discussed earlier? And we show that in the infected mice, in the salivary gland, 1 out of 6 of the mice actually develop focus score. When the lacrimal gland, you see 5 out of 6 mice actually develop focus score in the lacrimal gland. Okay, so next we examine the ANA profile. As you can see here, when we compare both males and female mice, we see an increase from 30% in the healthy control mice. When infected mice, you see an increase in 70% of ANA positive signal. In the male mice, you see, my apology, I cannot see it's too small for me to see here, but I'll go to my slide here. Maybe it's bigger, I can see. Yes. In the male mice, you see an increase from 20 to 80%. In the female mice, you see 40 to 70% of ANA being positive. Then we look at some of the antibody that's associated with Sjögren's. You see that there's an increase in the SSB or LAW antibodies, right, in the infected mice. And that, we look at male and female, they all look the same. And you see no significant changes with the SS row 52 and row 60 in the mouse infected with Sjögren. And then this is a collaboration with my colleagues at NCR, Dr. Blake Warner. He actually have recruited about a number of patients that have convalescent COVID-19. And so far, the study we published have 8 patients there. And this is a 6 out of 8 patient we look at. And you can see, my apology, but the thing, so I'm gonna look at my other screen where I can see much bigger here. So these are the 6 patients that we look at, the different ages here, right? And you look at the last column where it shows biopsy post COVID-19. This is months after COVID-19 diagnosis, right? And you can see patient one, 8 months. Patient two, you have, I think it's 3 days after diagnosed with COVID-19 and 6 months after. And then patient three, 13 months, 21 months. And then six is 7 months after diagnosed with COVID-19. But you look at the pathology, look at the histology, the clinical features here, right? They all develop some sort of adenitis in the salivary gland. I mean, they have inflammations in the salivary gland. Look at the focus score. They have a variety of changes in focus score, but look at patient number two. It's actually 3 weeks, 3 days post COVID-19, the individual has a focus score of 1. 6 months after, has a focus score of 2. So that's actually a chronic, um, chronic inflammation here that involve an increase in focus score in these patients. And you can see they actually develop different symptoms of ocular and symptom signs that we all experience. And one amazing is that this is only 6 out of 8 patients, and the 6 patients, they all exhibit some sort of Sjögren-like symptoms after infection with SARS-CoV-2. So this is an example of what they looked at when we look at histological changes here. Um, Here we look at that you have the healthy control, HV for healthy control, then you have P1, 2, 3, 4, 5, and 6. And you can see histologically, they have some of the classic features of Sjogren's, right? So this is post-convalescent COVID-19 subjects. You have infiltrate, you have focus score in the minor lip biopsies, you have fibrosis in the minor lip biopsies. So clearly there's a attractions of lymphocytes into the glands, and there's lead to destruction of the gland by fibrotic tissues. And SJD are a example of what a partial control should look like. So one thing that they looked at, but were positive, is that none of the patient they look at, the 8 patient they looked at, have ANA positive. So because of that, we have a cohort at University of Florida here, and we want to see what the profile looks like. Here, we look at I think 20 controls and 20 patients with COVID-19, and you can see that where you see the control here, you see if it's about 30% homogeneous staining using the help2 cells, right? And then you have 70% of negative. When you actually look at patient with COVID-19, the NA change from, I think this is about 70% to 30%. So 70% positive, from 30% negative from 30% positive to 70% positive when you have COVID-19. Then we look at the classic Sjögren associated antibodies. Look at Rho52, Rho60. We look at LAW, and you can see that a patient with COVID-19 actually elevated Rho52 and LAW antibodies in the serum. The other thing that we looked at is the two reasons for this. We actually made monoclonal antibodies from patients with SARS-CoV-2. Okay, the reason for that is we can use antibody for treatment, and we want to see if they can be those antibodies that specific against SARS-CoV-2. Can they also recognize nuclear antigens? And you can see here on A here is we look at inhibition of this monoclonal antibody we isolated. And you want to look at the A10, B5, and SA here, right? A10 and B5 have the best inhibition of viral infection, okay? When we screen for pseudovirus. Then we look in the bottom when we look at actually the plaque assay, the PRNT assay, and you can see that A10 has the least amount of plaque. That means they have the best inhibitions of viral infection, okay? Then we took those monoclonal antibodies and we stained and helped to cell look for autoantigens here. If you look at the first one, we will see A10, right, and B5 and SA. So A10 and B5 have the best inhibition, and they also show the best nuclear stain in there, where S8, which has the least inhibition, you don't see much of the positive for anti-nuclear antigens. So showing that the antibodies that we isolate have the best inhibition, actually the one that recognize the autoantigens. So that's an interesting fact that we need to explore further. Okay, so in summary here, SARS-CoV-2 infection in mice and human, the virus can persist in the lacrimal gland and the thymus gland. In the mice and the human, we have different pathology associated with different diseases. But in mouse, you see salivary flow loss, ANA, you see SSB, you see salinitis and dacryoanitis. In human, you see positive ANA, SSB, ROV2, and we see chronic salinitis in their biopsies. So, so what happened next is how do we treat these patients? So convention in my laboratory for the last 3 or 4 years, we're looking at how we can actually modulate the immune system at the antigen presentation part of it. Okay, so we look at the HOA or the MSC molecule, and we actually use an in silico screening to actually to determine which of this repurposed drug can actually block this HOA. And here is an example of the IAB HOA MSC from the mice that we generated, the AC1C2, and have an IAB haplotype. Here we identify 3 drugs that can bind to this IAB MSC, right? Buformin, metformin, and CHPE. We actually screen this using ELISA-based assay, and you can show that actually inhibit T cell function by decrease in IL-2 secretion. And then B is actually asking, okay, so we have the drugs that can inhibit B cell, T cell activation. Is it an antigen specific manner? So what happens when we actually generate TCR, T cell receptor T cells, T cell receptor cells, right? That have T cells that only recognize the rho 52, rho 60, and law. Okay, and then what we do is we incubate those TCR transduced T cells with the antigen presented cells, and then add the rho 52, rho 60, and law. Also, we add the drugs to see if they can suppress the proliferation of those TCR. As you can see there, when we add rho 52, rho 60, law with CHP and metformin, you can see a decrease in the proliferation of the TCR transfected cells. So it's shown that this blocking actually antigen specific, right? So is that true in the mouse model? We actually tested the drug in the mouse model we developed. And you can see that that when we treat the mouse with this drug, metformin, CHP, actually increase saliva flow rate in this mice. And there's a group in Korea that have shown that metformin, which is a drug for treating Taiwan diabetes, for treating lupus, actually shown to be very effective in treating NOD mouse model. So the question is, what I mentioned is those are chronic Sjögren syndrome disease. What about the COVID-19 induced associated Sjögren syndrome? How do you treat them? I think this is a very complicated question, and I think I have a schematic of how we can address that. I don't have any. I think it needs more research here. But in terms of, we have to be diagnosed whether this is a SARS-CoV-2 induced infection or this is a chronic infection that not related to SARS-CoV-2. We have to be diagnosed that properly. The question is, are we going to treat the patient with antiviral drugs, or are we going to treat that patient with autoimmune management drug like corticosteroid, anti-suppressant, biologics, immune suppressant, sorry. So this is a question that we have to really understand. And then we can monitor the viral load. We can monitor the changes in the clinical outcomes, right? And how does that translate to change in your lifestyle so you can improve your clinical outcomes. So in conclusion, I think having a comprehensive chemokine cytokine panel enable us to explore new novel pathways associated with Sjögren and so identify similarities that we see in the cytokine signal with long COVID. It's for us is for clinicians it's crucial to differentiate whether a chronic autoimmune disease of Sjögren is induced by SARS-CoV-2 or some sort of genetic aspect of it that lead to disease, and how we can treat that effectively, tailor the treatment to treat patients with those. And so that's my conclusion. This is the lab. The two people that actually work most on the project, my student Duran Shen and my biological scientist Alexandria. They involved in all the testing of SARS-CoV-2 in the mouse model. I want to thank my collaborator at NDCR, especially Dr. Warner, in terms of sharing data with us that we can publish and then learn more about long COVID and Sjögren's. And I want to thank you NDCR for their support. Well, thank you so much. Please, if you have any questions, let me know.
Dr. Nguyen, thank you for that excellent presentation. It is now time for our live Q&A portion of our webinar. So to our audience, as a reminder, if you have any questions you'd like to ask, please do so now. We have quite a few that have already come in, but if you have more questions, just type them into the Ask a Question box, click Submit, and we will answer as many of your questions as we have time for. I do want to bring your attention to the far left of your screen. We also have some survey questions for you. So your participation is greatly appreciated. And above that survey, you will see some resources that will help you get some more information. So Dr. Nguyen, let's dive right in. I'm just going to start from the top here. Very interesting talk. I am curious to know why these autoantibodies target only the salivary and tear glands exclusively.
Oh, so I think this is a great question. So it's not. So we have done for the purpose of Sjögren, yes, that we look at antibodies that target the salivary gland. We actually did a whole proteomic approach using antibody from SARS-CoV-2 patients, and I think they can actually recognize like 57 different antibodies. Okay, in terms of the cellular response, the cells, the different proteins in the cell, of systemic cell. So so it's not specific to the lacrimal salivary gland proteins, but I think it's a systemic.
And Dr. Nguyen, can the Sjögren autoantibodies be specifically removed from patients as intervention measures?
Ooh, I think this is a great, yes. I was thinking the same thing when I look at the data we generated in terms of monoclonal antibody we isolated, right? So we learned that, yeah, they can actually recognize nuclear antigens, but they actually also spike protein specific. So we have never done the experiment, but my guess is yes. Well, you have to be careful though. I think you have to really, you have to be virus specific too. So what we done is that we actually screen for antibodies that recognize the spike protein, right? And then we test whether they recognize the nuclear antigens or not. So can someone just take blood serum from COVID-19 patients and use a treatment? May or maybe not.
Okay, next question. In acute and severe COVID patients, most of all the above mentioned cytokines were unregulated. Did they also exhibit Sjögren's syndrome-like manifestations similar to LC patients?
What is LC patients? Do they exhibit children like? So, well, let me answer the first part. The second part, we don't have to get to that. So to answer the question, I do not know what people have done. So that for us is we look at patients with convalescent cognitive patients and Sjögren, but I think a lot of studies don't look at that, right? They look at severe or acute COVID-19 patient. They don't look at the autoimmune response of it. They may look at antibodies profile, right? but they actually never actually correlate to actual Sjögren. So to answer the questions, I don't know the answer for that. In my study, yes, but in other studies, I do not know.
LC being long COVID.
Oh, I see, I see.
Okay. Jumping to our next question. You mentioned the mouse kit contained many cytokine and chemokines. Were you able to identify new or further characterize specific proteins and pathways?
Good question. So I told you we learned a few that we've never seen in Sjögren, for example. I don't know what it does. We learned this. I think we learned that. I can go back to my, and I look up like, have anybody look at this, and no one has. So, uh, so we learned that, for example, IL-33. I don't know what it does in Sjögren. Uh, I learned that IL-3 it's actually highly expressed in um, IL-3 is highly expressed in all mice with Sjögren. I don't know what it does. You know, so these are great things I need to explore. We learned some of the chemokine receptor here, so we know that some of the chemokines are overexpressed. For example, we learned that CXCL2, CXCL1, right? They involve in recruitment of memory T cells, but what else does it do? I do not know. There's, in terms of how does it correlate to actual human patients, I do not know. So I think we learn a lot from this comprehensive panel. Yes.
So, Dr. Nguyen, for the chemokine and the cytokine levels in the mice, have you observed expression patterns over time? Are there any differences between expression and disease severity or progression in the mouse models?
So yes, yes. So for the study, we look at young mice and old mice in the healthy controls and the disease control mice, right? You can clearly see that there are a lot of cytokines or chemokines that it's actually highly upregulated in the disease stage, in the disease mouse, in the older mouse, and it's not. It's actually down in the younger mice. So there are clearly changes between that age where I think when we look at the young mice, we define at 0 to 8 weeks of age, and then the older mice are like 24 to 35 weeks of age. So clearly, there are cytokine level that we can measure at that two stages that really tell us, the disease process, especially the. I'm interested in the recruitment of all the lymphocytes in the salivary glands and lacrimal gland. So yes. Yes.
Dr. Nguyen, in this presentation, Sjogren's is associated with COVID infection. Have you observed the same post-mRNA COVID vaccines?
No, we did not. No, we did not. No, we did not. No, no.
Okay. So it is already recommended that dexamethasone and other systemic corticosteroids to treat outpatients with mild to moderate COVID-19. So what is the value of your autoimmune management for COVID-19 associated SJD flowchart out of the current treatment scheme?
Okay, I'm gonna read this question so I can. So this is interesting, right? Because you are correct that dexamethasone and other corticosteroids are used to treat both COVID-19 symptoms and also to treat autoimmune diseases. Um, I, so first of all, I don't think maybe some patients are more responsive to drugs than others, but it's not a really good. To me, what I've seen is that it's not a very good treatment, right? Uh, Also, it's not a in terms of its relationship to the severity of COVID-19, right? I don't think dexamethasone and corticosteroid can actually uh, alleviate symptoms associated with the different severity of COVID-19, right? Um, in terms of patient with Sjögren, although that's been used, I'm not sure that's like a to me that's a very it's to alleviate some of the extracellular symptoms it's not to treat patient with Sjögren. So I agree with you on that aspect of it, but I think we really need to look at whether it's a viral induced, right, where you actually treat the virus, get rid of the virus, and instead of just treating the clinical aspect of it, the after effect of viral infection. So we have to look at that and how we can treat patients better.
Dr. Nguyen, are we talking primary or secondary Sjögren's?
Okay, so the primary okay. So I think for the mouse, we are only look at the primary Sjögren syndrome, right, which I didn't discuss. We are only look at the changes in the lacrimal and the salivary gland. We couldn't ask the mice, what do you feel depressed? We couldn't ask those systemic. We always we didn't look at. We look at the brain. We look at the lungs. We see some changes in that, but we don't see drastic change in the brain or other part of the. We don't see infiltrates in the kidney, right? That we probably some seen in patient with long COVID. So for our mouse study, we look at the primary aspect of it. For secondary Sjögren, we did not look, and I don't want to get overemphasized on that aspect of it.
Thank you. Now, how do you measure salivary secretion?
This is a great question. I love answering this question. Okay, so so what we do is we inject the mouse with isoproterenol and pilocarpine to induce their secretion. Um, and then what we do is you take the mouse. Here, actually, I have a mouse here. You take the mouse like this, right? And you tear it one way like that, and then you take a pipette with a pipette tip, and as they will salivate on the edge of their mouth here, and then we collect every drop of the saliva and put in a test tube. So if you have 100 mice, so each mouse will take us 11 minutes to collect. So my lab members are great at sitting, listen to music, and then collect mouse saliva. So if we have 100 mouse, that would take 100 times 11. That's a lot of minutes. So that's what we do. Every mouse. And then we measure how much it actually secrete. That's how we measure saliva secretion.
Definitely time consuming, but. So, Dr. Nguyen, could you tell us, please, also if this is an autoimmunological disease and should we treat it with autohistamine drugs?
Okay. So a colleague of mine actually here have found that some antihistamine can actually suppress SARS-CoV-2. Okay, in terms of I think they did both in vivo and in vitro. Okay, now in terms of the autoimmune disease, I don't want to speculate that, but I know what he done. He published that study. David Astro. They show that antihistamine can actually prevent the infection of SARS-CoV-2. Now, in terms of actually treating Sjögren's with SARS that is induced by SARS-CoV-2, I don't think I want to venture to that part of it. So I think to be determined. Great question, though. Yes.
CBD. Okay. Is there an association between the severity of COVID-19 and the likelihood of developing Sjögren's syndrome post-infection?
So in the human data, I think we need more. We need more patients to actually to answer that question. It's a great question. In the mouse, however, we learned this. When we infect ANN60 peer review of the Wuhan strain, for 21 days, they develop what we've seen here, right? We actually using the we tried the Delta strain and the Omicron strain in this mouse model. They actually do not. We use up to 1,000 peer review, right? So they do not develop the same symptoms that we see in the Wuhan strain. So to answer the question, I think the severity of uh, severity both of COVID-19 and Sjögren is actually depends on the dosage of the virus and the variant of the virus. I think that's critical here. Um, that we need to explore more. I don't have an answer for that, but from what we've done in the lab, we learned that yes, the severity of COVID-19 is it, and the severity of Sjögren is actually virus dose dependent and the variant dependent. I think we need to look at that more.
Excellent. Dr. Nguyen, we have a lot of questions coming in, but we're going to squeeze 2 more in here before we run out. Which viral factors are likely to increase the probability of developing Sjogren's syndrome symptoms following SARS-CoV-2 infection?
So that's a great question. And I think also we need to learn more about this. But for us is that we found there is a persistent viral infections, persistent viral particles in the salivary gland, right? So it's there, 21 days post-infection, okay? The other things that we learned is that they are there for a reason, and they actually are there to actually lead to apoptosis of some of the ductal cells and actin cells in the glands, right? And this is like the pre, when we learn about the pre-disease onset, right? When you have that wave of apoptosis in the gland, and it's not controlled, okay? Then you have influx of you activate chemokine, cytokine, and influx of immune cells in there that allow that activation. So to answer the questions, I think the main with us is this is the viral persistent that lead to apoptosis, right? And that actually, when the apoptosis happen, you have a so like a leakage of all the cellular proteins, right? And now the immune system see this as a something they can now immune response again. This is also how you make so much antibodies with patient with SARS-CoV-2, um, COVID-19 infection.
And Dr. Nguyen, we're going to wrap with this final question. Were cytokine and chemokine profiles also measured in the salivary glands and tears of the animal models? If so, how do these profiles correlate with plasma levels?
So for this study, we look at the chemokine cytokines only in the sera. We did not look at the cytokine chemokine levels in tears and saliva. I think we need to look at that. And I guess I didn't make that clear. We look at only in sera. Now, in previous study where we actually correlate the actual cytokine secretions in saliva and blood, and they actually correlate quite well. For example, when we look at all the cytokines involving Th17 cells like IL-17, IL-21, 27, they actually correlate in terms of uh, the amount. Actually, let me take it back. I don't think the IL-17 is it's found mostly in there's few things we look at. We look at cell lysate. So I gotta be clear. So your question is asking whether the tear saliva correlate with the plasma. I think the answer is yes. Do they correlate with the actual cell vegan lysate? It may or may not, depending on which cytokine you look at.
Excellent. Dr. Nguyen, thank you so much for your time today and your important research. Do you have any final comments you'd like to leave our audience with?
No. Oh, actually, I do. I think so far, I thank you, the audience, for all the questions here. Great questions. These are questions that we are thinking doing the lab right now, and the questions that we think about the future of doing. So I love the audience today and how the questions are quite inquisitive, and I enjoy today a lot. I appreciate you, Christy, and the audience for being here today.
Well, we appreciate you and your important information. This is so helpful. I do want to thank LabRoots and our sponsor, Olink Proteomics, for underwriting today's educational webcast. Before we go, I want to thank our audience again, too, for joining us today and for their interesting questions. Questions we did not have time for today, and there were quite a few, and those that are submitted during the on-demand period, they will be addressed by Dr. Nguyen via the contact information you provided at the time of registration. Today's webcast will also be available for on-demand viewing, and Laboratory will alert you via email when that webcast is available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. And if you missed filling out that survey, it will pop up on your screen when we close. To all our friends out there, thank you for joining us. Have a great day, everyone. Bye-bye.
Thank you so much.